Let's look more in depth at how we carry oxygen throughout our body in the blood. We have a picture here. These blue dots represent oxygen within an alveolus, and we have the capillaries that are surrounding the alveoli. So there's a dense number of capillaries surrounding the alveoli to help us carry oxygen. So oxygen is lipophilic, so it moves very readily across the alveolar cells as well as the capillary endothelial cells and makes its way into plasma. Now, this amount of oxygen that's dissolved in plasma is really not very much at all. So let's look at how much oxygen our plasma can carry. If we can consider that we have about 5 liters of blood in our body, the amount of oxygen that can dissolve per liter of blood is only about 3 milliliters of oxygen. So with our 5 liters of blood, that gives us a carrying capacity of about 15 milliliters of oxygen. Well, to put that in perspective, it takes about 250 milliliters of oxygen per minute at rest to meet our basic metabolic needs. Now, the addition of hemoglobin greatly enhances our ability to carry oxygen. So we have our baseline 3 milliliters of oxygen dissolved in the plasma, but when we add hemoglobin, notice that we now can carry much more oxygen. And this actually increases the amount of oxygen by about 200 milliliters of oxygen per liter of blood. So taking our 5 liters of blood, we now have over 1,000 milliliters of oxygen that we can carry. So that's more than enough to meet our basic needs of 200 milliliters of oxygen per minute. When we measure the partial pressure of oxygen, realize though that that only reflects the oxygen that's dissolved in plasma and doesn't reflect the amount of oxygen that's bound to hemoglobin. And we'll see that on this next slide. This is a graph which we'll talk more about in a couple of slides, but this is a picture of the oxygen-hemoglobin saturation curve. On the x-axis, we have the partial pressure of oxygen, and on the y-axis, we have the percent saturation of hemoglobin. So the y-axis reflects all the oxygen present and the percentage of hemoglobin that's saturated. The PO2, however, only represents the oxygen that's dissolved in plasma. But in reality, this is really what's important, is the oxygen that's dissolved, because this is the oxygen that's going to be able to diffuse over into the tissues. So the PO2 is the most important clinical number. Now, obviously, the PO2 and O2 sat are related to each other, but the PO2 only measures the oxygen dissolved in plasma. So let's look at how oxygen actually is transmitted or transported throughout our body. So we have an alveolus here, we have our tissues here, and then we have our systemic circulation arterial side shown here in red and the venous side in blue. So the oxygen, if we were to measure the oxygen in the alveoli, we would find that it's about 100 millimeters of mercury in the alveoli. So that oxygen in the tissue, the partial pressure of oxygen in the tissue, is only about 40 millimeters of mercury. So when we have oxygen in the alveolus, realize that it's going to diffuse out of the alveolus into the pulmonary capillary, and we show that small amount that's going to be dissolved in plasma. We also are going to have oxygen that is going to be diffusing into the plasma and then subsequently into the red blood cells and binding to hemoglobin. So with this movement of oxygen into the blood, this will end up giving us a PO2 of about 100. So this is fully saturated hemoglobin taking blood now from the lungs to the tissues. So blood is going to be carried to the tissues, and realize that at the tissues we're going to go from a PO2 of 100 in the blood that's flowing through, and it's going to be taking it to tissues which have a PO2 of about 40. So as a result, this blood, which has a PO2 of 100, we now have a fairly significant gradient here. So oxygen then will move from the blood into the tissues. And recall that that initial movement is from the O2 that's dissolved in plasma. And then as that O2 that's dissolved in the plasma moves out to the tissue, hemoglobin then will release its oxygen into the plasma, and that subsequently can then be taken to the tissue or delivered to the tissues. So this process of having the oxygen leave the blood and move to the tissues, we call that unloading of oxygen. And once we're through unloading the oxygen, we'll find that the blood that leaves that tissue has a PO2 down only about 40. And this then is going to be taken back to the lungs, where it will then go through this cycle once again. Now back to our oxygen hemoglobin saturation curve. I've added a few additional lines, and this is showing that in the arterial side, our PO2 is about 100. And with that, our hemoglobin is nearly 100% saturated. When we then take the blood to the tissues and drop off our oxygen, we go down to a PO2 of 40. That leaves our hemoglobin about 75% saturated or so. Now this curve reflects the binding of hemoglobin to oxygen. So realize this is a ligand binding to a binding site. So this is a curve which is reflecting the affinity of oxygen and hemoglobin. This is a curve that we might switch. We might shift this curve to the right, or we may shift the curve to the left. So realize that by shifting the curve, we are reflecting a change in affinity of hemoglobin for oxygen. A shift of the curve to the right represents a decrease in the affinity of hemoglobin for oxygen, and a shift to the left represents an increase in affinity of hemoglobin for oxygen. So what kind of things are going to cause this curve to shift? An easy way to remember this is thinking about what causes a shift to the right, which would be a decrease in affinity, and that is going to be things that are associated with exercise or exertion. So an increase in temperature. Metabolically active tissues have heat that's generated, so an increase in temperature will shift this curve to the right. An increase in hydrogen ions, or stated another way, a decrease in pH, also associated with metabolic activity, shifts the curve to the right. An increase in CO2 produced by metabolic pathways shifts the curve to the right. This particular shift is called the Bohr Effect. And then there's another substance called 2,3-BPG or 2,3-DPG. This is an intermediate of glycolysis. So when our tissues are metabolically active, glycolysis is happening at a higher rate, producing this substance, and all of these substances have the same effect of shifting the curve to the right. So realize what this reflects. During exercise, hemoglobin has a reduced affinity, and we can show that by this curve shifting to the right. So notice that our PO2 at the arterial side doesn't change because that's just hemoglobin that's been saturated as it goes through the lungs. But when we go to the tissues, notice the difference of changing affinity. At our baseline here, we said that we left the tissues and went back into the veins with a hemoglobin saturation of about 75% or so. But notice with a shift in the curve to the right, we are not saturated at 75%, we're now down maybe around 50% or less. So this shift to the right reflects that we are unloading more oxygen at the tissues. And that's exactly what we want to happen during exercise.